Electron transport chain, Wikipedia audio. An electron transport chain is a series of complexes that transfer electrons from electron donors to electron acceptors via redox reactions, and couples this electron transfer with the transfer of protons across a membrane. This creates an electrochemical proton gradient that drives the synthesis of adenosine triphosphate, a molecule that stores energy chemically in the form of highly strained bonds. The molecules of the chain include peptides, enzymes, and others. The final acceptor of electrons in the electron transport chain during aerobic respiration is molecular oxygen, although a variety of acceptors other than oxygen such as sulfate exist in anaerobic respiration. Electron transport chains are used for extracting energy via redox reactions from sunlight in photosynthesis or, such as in the case of the oxidation of sugars, cellular respiration. In eukaryotes, an important electron transport chain is found in the inner mitochondrial membrane, where it serves as the site of oxidative phosphorylation through the use of ADP synthase. It is also found in the thylakoid membrane of the chloroplast in photosynthetic eukaryotes. In bacteria, the electron transport chain is located in their cell membrane. In chloroplasts, light drives the conversion of water to oxygen and NADP and to NADP with transfer of H and ions across chloroplast membranes. In mitochondria, it is the conversion of oxygen to water, NADH to NAD and N-succinate to fumarate that are required to generate the proton gradient. Background: electron transport chains are major sites of premature electron leakage to oxygen, generating superoxide and potentially resulting in increased oxidative stress. The electron transport chain consists of a spatially separated series of redox reactions in which electrons are transferred from a donor molecule to an acceptor molecule. The underlying force driving these reactions is the Gibbs free energy of the reactants and products. The Gibbs free energy is the energy available to do work. Any reaction that decreases the overall Gibbs free energy of a system is thermodynamically spontaneous. The function of the electron transport chain is to produce a transmembrane proton electrochemical gradient as a result of the redox reactions. If protons flow back through the membrane, they enable mechanical work, such as rotating bacterial flagella. ADP synthase, an enzyme highly conserved among all domains of life, converts this mechanical work into chemical energy by producing ADP, which powers most cellular reactions. A small amount of ADP is available from substrate-level phosphorylation, for example, in glycolysis. In most organisms, the majority of ADP is generated in electron transport chains, while only some obtain ADP by fermentation. Most eukaryotic cells have mitochondria, which produce ADP from products of the citric acid cycle, fatty acid oxidation, and amino acid oxidation. At the mitochondrial inner membrane, electrons from NADH and FADH2 pass through the electron transport chain to oxygen, which is reduced to water. The electron transport chain comprises an enzymatic series of electron donors and acceptors. Each electron donor will pass electrons to a more electronegative acceptor, which in turn donates these electrons to another acceptor, a process that continues down the series until electrons are passed to oxygen, the most electronegative and terminal electron acceptor in the chain. Passage of electrons between donor and acceptor releases energy, which is used to generate a proton gradient across the mitochondrial membrane by actively pumping protons into the intermembrane space, producing a thermodynamic state that has the potential to do work. The entire process is called oxidative phosphorylation, since ADP is phosphorylated to ADP using the energy of hydrogen oxidation in many steps. A small percentage of electrons do not complete the whole series and instead directly leak to oxygen, resulting in the formation of the free radical superoxide, a highly reactive molecule that contributes to oxidative stress and has been implicated in a number of diseases and aging. Energy obtained through the transfer of electrons down the etc. is used to pump protons from the mitochondrial matrix into the intermembrane space, creating an electrochemical proton gradient across the inner mitochondrial membrane. This proton gradient is largely but not exclusively responsible for the mitochondrial membrane potential. It allows ADP synthase to use the flow of HN through the enzyme back into the matrix to generate ADP from adenosine diphosphate and inorganic phosphate. Complex I accepts electrons from the Krebs cycle electron carrier nicotinamide adenine dinucleotide, and passes them to coenzyme Q, which also receives electrons from complex II. Q passes electrons to complex III, which passes them to cytochrome C. CytC passes electrons to complex IV, which uses the electrons and hydrogen ions to reduce molecular oxygen to water. 4 membrane-bound complexes have been identified in mitochondria. Each is an extremely complex transmembrane structure that is embedded in the inner membrane. 3 of them are proton pumps. The structures are electrically connected by lipid-soluble electron carriers and water-soluble electron carriers. The overall electron transport chain. In complex I, 2 electrons are removed from NADH and ultimately transferred to a lipid-soluble carrier, ubiquinone. The reduced product, ubiquinol, freely diffuses within the membrane, and complex I translocates 4 protons across the membrane, thus producing a proton gradient. Complex I is one of the main sites at which premature electron leakage to oxygen occurs, thus being one of the main sites of production of superoxide. The pathway of electrons is as follows. In mitochondria, NADH is oxidized to NAD+, by reducing flavin mononucleotide to FMNH2 in 1,2 electron step. FMNH2 is then oxidized in 2 1 electron steps, through a semiquinone intermediate. Each electron thus transfers from the FMNH2 to an FES cluster, from the FES cluster to ubiquinone. Transfer of the first electron results in the free radical form of Q, and transfer of the second electron reduces the semiquinone form to the ubiquinol form, QH2. During this process, 4 protons are translocated from the mitochondrial matrix to the intermembrane space. As the electrons become continuously oxidized and reduced throughout the complex, an electron current is produced along the 180 angstrom width of the complex within the membrane. This current powers the active transport of 4 protons to the intermembrane space per 2 electrons from NADH. This complex is inhibited by alkyl guanides, rotenone, barbiturates, chlorpromazine, pyricidine. In complex 2 additional electrons are delivered into the quinone pool originating from succinate and transferred to Q. Complex 2 consists of 4 protein subunits: succinate dehydrogenase, succinate dehydrogenase iron sulfur subunit, mitochondrial, succinate dehydrogenase complex subunit C, and succinate dehydrogenase complex, subunit D. Other electron donors also direct electrons into Q. Complex 2 is a parallel electron transport pathway to complex 1, but unlike complex 1, no protons are transported to the intermembrane space in this pathway. Therefore, the pathway through complex 2 contributes less energy to the overall electron transport chain process. This complex is inhibited by carboxyl. In complex 3, the Q cycle contributes to the proton gradient by an asymmetric absorption/release of protons. 2 electrons are removed from QH2 at the QO site and sequentially transferred to 2 molecules of cytochrome C, a water-soluble electron carrier located within the intermembrane space. The 2 other electrons sequentially pass across the protein to the QI site where the quinone part of ubiquinone is reduced to quinol. A proton gradient is formed by 1 quinol oxidations at the QO site to form 1 quinone at the QI site. When electron transfer is reduced, complex 3 may leak electrons to molecular oxygen, resulting in superoxide formation. This complex is inhibited by British antylylysate, naphthoquinone, and antimycin. Mitochondrial redox carriers. Complex I. In complex 4, sometimes called cytochrome AA3, 4 electrons are removed from 4 molecules of cytochrome C and transferred to molecular oxygen, producing 2 molecules of water. At the same time, 8 protons are removed from the mitochondrial matrix, contributing to the proton gradient. The activity of cytochrome C oxidase is inhibited by cyanide, carbon monoxide, azide, hydrogen sulfide. Complex 2. Complex 3. Complex 4. Coupling with oxidative phosphorylation. Summary. According to the chemiosmotic coupling hypothesis, proposed by Nobel Prize in Chemistry winner Peter D. Mitchell, the electron transport chain and oxidative phosphorylation are coupled by a proton gradient across the inner mitochondrial membrane. The efflux of protons from the mitochondrial matrix creates an electrochemical gradient. This gradient is used by the FOF1 ADP synthase complex to make ADP via oxidative phosphorylation. ADP synthase is sometimes described as complex V of the electron transport chain. The FO component of ADP synthase acts as an ion channel that provides for a proton flux back into the mitochondrial matrix. It is composed of A, B and C subunits. Protons in the intermembranous space of mitochondria first enters the ADP synthase complex through a subunit channel. Then protons move to the C subunits. The number of C subunits it has determines how many protons it will require to make the FO turn one full revolution. For example, in humans, there are 8 C subunits, thus 8 protons are required. After C subunits, protons finally enters matrix using a subunit channel that opens into the mitochondrial matrix. This reflux releases free energy produced during the generation of the oxidized forms of the electron carriers. The free energy is used to drive ADP synthesis, catalyzed by the F1 component of the complex. Coupling with oxidative phosphorylation is a key step for ADP production. However, in specific cases, uncoupling the two processes may be biologically useful. The uncoupling protein, thermogenin present in the inner mitochondrial membrane of brown adipose tissue provides for an alternative flow of protons back to the inner mitochondrial matrix. Thyroxine is also a natrial uncoupler. This alternative flow results in thermogenesis rather than ADP production. Synthetic uncouplers also exist, and, at high doses, are lethal. In the mitochondrial electron transport chain, electrons move from an electron donor to a terminal electron acceptor via a series of redox reactions. These reactions are coupled to the creation of a proton gradient across the mitochondrial inner membrane. There are 3 proton pumps, I, 3, and 4. The resulting transmembrane proton gradient is used to make ADP via ADP synthase. The reactions catalyzed by complex I and complex 3 work roughly at equilibrium. This means that these reactions are readily reversible, by increasing the concentration of the products relative to the concentration of the reactants. ADP synthase is also readily reversible. Thus ADP can be used to build a proton gradient, which in turn can be used to make NADH. This process of reverse electron transport is important in many prokaryotic electron transport chains. In bacteria. In eukaryotes, NADH is the most important electron donor. The associated electron transport chain is NADH complex IQ complex 3 cytochrome C complex 4O2, where complexes I, 3 and 4 are proton pumps, while Q and cytochrome C are mobile electron carriers. The electron acceptor is molecular oxygen. In prokaryotes the situation is more complicated, because there are several different electron donors and several different electron acceptors. The generalized electron transport chain in bacteria is. Note that electrons can enter the chain at 3 levels: at the level of a dehydrogenase, at the level of the quinone pool, or at the level of a mobile cytochrome electron carrier. These levels correspond to successively more positive redox potentials, or to successively decreased potential differences relative to the terminal electron acceptor. In other words, they correspond to successively smaller Gibbs free energy changes for the overall redox reaction donor acceptor. Individual bacteria use multiple electron transport chains, often simultaneously. Bacteria can use a number of different electron donors, a number of different dehydrogenases, a number of different oxidases and reductases, and a number of different electron acceptors. For example, E. coli uses 2 different NADH dehydrogenases and 2 different quinol oxidases, for a total of 4 different electron transport chains operating simultaneously. A common feature of all electron transport chains is the presence of a proton pump to create a transmembrane proton gradient. Bacterial electron transport chains may contain as many as 3 proton pumps, like mitochondria, or they may contain only 1 or 2. They always contain at least 1 proton pump. In the present-day biosphere, the most common electron donors are organic molecules. Organisms that use organic molecules as an energy source are called organotrophs. Organotrophs and phototrophs constitute the vast majority of all familiar life forms. Electron donors. Some prokaryotes can use inorganic matter as an energy source. Such an organism is called a lithotroph. Inorganic electron donors include hydrogen, carbon monoxide, ammonia, nitrite, sulfur, sulfide, manganese oxide, and ferrous iron. Lithotrophs have been found growing in rock formations thousands of meters below the surface of Earth. Because of their volume of distribution, lithotrophs may actually outnumber organotrophs and phototrophs in our biosphere. The use of inorganic electron donors as an energy source is of particular interest in the study of evolution. This type of metabolism must logically have preceded the use of organic molecules as an energy source. Dehydrogenases. Bacteria can use a number of different electron donors. When organic matter is the energy source, the donor may be NADH or succinate, in which case electrons enter the electron transport chain via NADH dehydrogenase or succinate dehydrogenase. Other dehydrogenases may be used to process different energy sources: formate dehydrogenase, lactate dehydrogenase, glyceraldehyde 3-phosphate dehydrogenase, H2 dehydrogenase, etc. Some dehydrogenases are also proton pumps, others funnel electrons into the quinone pool. Most dehydrogenases show induced expression in the bacterial cell in response to metabolic needs triggered by the environment in which the cells grow. Quinones are mobile, lipid-soluble carriers that shuttle electrons between large, relatively immobile macromolecular complexes embedded in the membrane. Bacteria use ubiquinone and related quinones such as menaquinone. Another name for ubiquinone is coenzyme Q10. Quinone carriers. Proton pumps. Cytochrome electron carriers. A proton pump is any process that creates a proton gradient across a membrane. Protons can be physically moved across a membrane. This is seen in mitochondrial complexes I and IV. The same effect can be produced by moving electrons in the opposite direction. The result is the disappearance of a proton from the cytoplasm and the appearance of a proton in the periplasm. Mitochondrial complex III uses this second type of proton pump, which is mediated by a quinone. Some dehydrogenases are proton pumps, others are not. Most oxidases and reductases are proton pumps, but some are not. Cytochrome BC1 is a proton pump found in many, but not all, bacteria. As the name implies, bacterial BC1 is similar to mitochondrial BC1. Proton pumps are the heart of the electron transport process. They produce the transmembrane electrochemical gradient that enables ADP synthase to synthesize ADP. Cytochromes are pigments that contain iron. They are found in two very different environments. Some cytochromes are water-soluble carriers that shuttle electrons to and from large, immobile macromolecular structures embedded in the membrane. The mobile cytochrome electron carrier in mitochondria is cytochrome C. Bacteria use a number of different mobile cytochrome electron carriers. Other cytochromes are found within macromolecules such as complex III and complex IV. They also function as electron carriers, but in a very different, intramolecular, solid-state environment. Electrons may enter an electron transport chain at the level of a mobile cytochrome or quinone carrier. For example, electrons from inorganic electron donors enter the electron transport chain at the cytochrome level. When electrons enter at a redox level greater than NADH, the electron transport chain must operate in reverse to produce this necessary, higher energy molecule. When bacteria grow in aerobic environments, the terminal electron acceptor is reduced to water by an enzyme called an oxidase. When bacteria grow in anaerobic environments, the terminal electron acceptor is reduced by an enzyme called a reductase. In mitochondria, the terminal membrane complex is cytochrome oxidase. Aerobic bacteria use a number of different terminal oxidases. For example, E. coli does not have a cytochrome oxidase or a BC1 complex. Under aerobic conditions, it uses two different terminal quinol oxidases to reduce oxygen to water. Anaerobic bacteria, which do not use oxygen as a terminal electron acceptor, have terminal reductases individualized to their terminal acceptor. For example, E. coli can use fumarate reductase, nitrate reductase, nitrite reductase, DMSO reductase, or trimethylamine N oxide reductase, depending on the availability of these acceptors in the environment. Most terminal oxidases and reductases are inducible. They are synthesized by the organism as needed, in response to specific environmental conditions. Just as there are a number of different electron donors, there are a number of different electron acceptors, both organic and inorganic. If oxygen is available, it is invariably used as the terminal electron acceptor, because it generates the greatest Gibbs free energy change and produces the most energy. In anaerobic environments, different electron acceptors are used, including nitrate, nitrite, ferric iron, sulfate, carbon dioxide, and small organic molecules such as fumarate. Since electron transport chains are redox processes, they can be described as the sum of two redox pairs. For example, the mitochondrial electron transport chain can be described as the sum of the NAD+/NADH redox pair and the O2/H2O redox pair. NADH is the electron donor and O2 is the electron acceptor. Not every donor-acceptor combination is thermodynamically possible. The redox potential of the acceptor must be more positive than the redox potential of the donor. Furthermore, actual environmental conditions may be far different from standard conditions, which apply to standard redox potentials. For example, hydrogen-evolving bacteria grow at an ambient partial pressure of hydrogen gas of 10⁴ atm. The associated redox reaction, which is thermodynamically favorable in nature, is thermodynamic impossible under standard conditions. Bacterial electron transport pathways are, in general, inducible. Depending on their environment, bacteria can synthesize different transmembrane complexes and produce different electron transport chains in their cell membranes. Bacteria select their electron transport chains from a DNA library containing multiple possible dehydrogenases, terminal oxidases, and terminal reductases. The situation is often summarized by saying that electron transport chains in bacteria are branched, modular, and inducible. In oxidative phosphorylation, electrons are transferred from a low-energy electron donor to an acceptor through an electron transport chain. In photophosphorylation, the energy of sunlight is used to create a high-energy electron donor and an electron acceptor. Electrons are then transferred from the donor to the acceptor through another electron transport chain. Photosynthetic electron transport chains have many similarities to the oxidative chains discussed above. They use mobile, lipid-soluble carriers and mobile, water-soluble carriers. They also contain a proton pump. It is remarkable that the proton pump in all photosynthetic chains resembles mitochondrial complex 3. Photosynthetic electron transport chains are discussed in greater detail in the articles Photophosphorylation, Photosynthesis, Photosynthetic Reaction Center and Light-Dependent Reaction. Electron transport chains are redox reactions that transfer electrons from an electron donor to an electron acceptor. The transfer of electrons is coupled to the translocation of protons across a membrane, producing a proton gradient. The proton gradient is used to produce useful work. About 30 work units are produced per electron transport. Terminal oxidases and reductases. Electron acceptors. Summary 2. Photosynthetic. Summary 3.